Revolution.
This is Brad Goodner. Welcome back to Genomics Revolution. In our first 6 episodes, we have introduced the terms genome and genomics, talked about how the field of genomics got its start, and looked at the steps of a genome project using the first ever sequenced cellular genome as an example. Now we will tour through a survey of some sequenced genomes. All 3 domains of life will be represented, but the bacteria and archaea will get the lion's share. For each genome, we will learn why scientists are interested in the organism, some basic data about the genome, its genes and encoded proteins, a few surprises from the genome sequence, and an example of how scientists have taken the next step past having the genome sequence. Each genome will be presented by a different student in the 2019 Hiram College Genetics course, and they will put their own unique spins on their assigned subjects. To get us started, I will talk about my favorite genome, number 50 by my count in terms of getting published. This is the genome of Agrobacterium tumefaciens strain C58, an organism I have worked on and off since 1983. Agrobacterium, or agro for short, is a genus from the bacteria division Alpha Proteobacteria found in soils all over the world, and is best known because some strains are plant pathogens. These pathogenic strains contain a plasmid that allows them to do something no other bacterial pathogens can do: transfer a piece of their own DNA into their eukaryotic host cell, where the expression of genes on the transferred DNA causes the cells of the plant host to act very, very differently. The transformed plant cells grow out of control because they make their own growth-stimulating hormones, and they produce and secrete some strange compounds that agro can use as carbon and nitrogen sources. It turns out that agro has been genetically engineering plants on its own for a long time, well before any humans thought about the possibility. I came back to work on Agrobacterium in 1996 when I read a paper by Alarde Servant et al., who showed that in strain C58, there are 2 DNA molecules greater than 1 million base pairs in size that contained ribosomal RNA genes. The presence of ribosomal RNA genes is usually indicative of a chromosome, but this would mean that strain C58 has 2 chromosomes, and there was no previous evidence of this. A group of 8 undergraduates at University of Richmond worked with me to generate and map a large collection of transposon insertions in essential genes of agro C58. Our paper, published in 1999, proved that there are, in fact, 2 chromosomes in strain C58. The larger chromosome is a circle of roughly 3 million base pairs, but the smaller 2.1 million base pair chromosome is a linear DNA molecule. Agro's closest relatives in the genus Cynorhizobium only show 1 circular chromosome of roughly 3.6 million base pairs. So we wondered, where did the smaller linear chromosome come from? We imagined 3 possibilities. 1. The smaller chromosome originated from a breakage event in the original circular chromosome. 2. The smaller chromosome came in from the outside, such as a viral infection. Or 3. Some combination of the first 2 hypotheses. It was this very question that drove my lab to start sequencing the agro C58 genome in 1999. We started with $3,000 to build a genomic library and start sequencing a few library clones. But we knew the full cost of this project would be closer to $500,000. In the fall of 1999, we presented some of our initial findings at a small research conference that focuses on the biology of Agrobacterium. At the end of the conference, a gentleman approached me. "My name is Steve Slater," he said, "and I work for a small company called Genomics. We need to talk, but not here. I will call you tomorrow." On the flight back home, I told my wife Asha that I thought Steve Slater was going to tell me that his group had already sequenced the C58 genome. However, the next day, Steve told me that Genomics was just beginning to sequence the genome and that they wanted to collaborate with my research team because of our genome map of transposon insertions. Steve rightfully saw the value of using our map to orient and join up the sequence pieces of the genome. We reached an agreement between Genomics, its parent company Monsanto Corporation, and my research lab. The agreement required all partners to agree as to when and how to publish the finished work. If any one partner didn't want to publish, the collaboration would stop. It was a fun but very odd collaboration. My students got to work with a lot more sequence information, but we had to use a dial-in modem connection on one computer to access the company database. This restriction slowed us down, but we made consistent progress and were basically finished with sequencing and assembling the genome sequence by the end of 2000. Around that time, we became aware of another collaboration between an academic lab at the University of Washington and DuPont Corporation that was also sequencing the same Agrobacterium C58 genome. It was a race now, but luckily in the end, both collaborations agreed to publish back-to-back articles in the journal Science that came out in December of 2001. So what did we learn from the Agro C58 genome sequence? First and foremost, the 2.1 million base pair linear chromosome was evolutionarily derived from a plasmid. You heard me right, a plasmid. The origin of replication on this chromosome is clearly a member of the Rep ABC plasmid family, very similar to those found on two large plasmids in strain C58. Second, the linearity of the second chromosome is due to hairpin loops on each end where the top and bottom strands are connected through a stem loop structure. In a later paper, we obtained the full sequence of the hairpin loops and showed that the linear chromosome is found only in one subset of Agrobacterium and Rhizobium strains called Barobar 1. During replication, the two so-called old strands are still connected at their ends. Once the hairpin loops are replicated, an enzyme called protelomerase recognizes the double-stranded hairpin sequences and makes staggered cuts, just like a restriction enzyme that generates an overhang. This allows the two new daughter double-stranded DNA molecules to separate and then reform single-stranded hairpin loops on each end. We don't know yet the evolutionary origin of the hairpin loops and the gene encoding protelomerase. My personal hypothesis is that they both came in together as part of a linear bacteriophage, but that will take more work in the future. The third big finding: comparison of the two chromosomes of Agro C58 with the sequenced single chromosome of its relative Cynorhizobium strain 1021 showed clear evidence that several large chunks of the ancestral circular chromosome had moved to the plasmid that became the second chromosome. I have had several Hiram College students continue studying this phenomenon, and this became part of yet another follow-up publication. There were a lot more insights gleaned from the Agro C58 genome sequence, and we continued to link genes to function using functional genomics experiments such as the Mariner type transposon mutagenesis screen going on in the 2019 Hiram College genetics course. However, I will leave those details for another time. The Agro C58 genome shows us how complex genomes can arise in the bacterial domain and how genomes can rearrange over time. Now let us see what we can learn from other genomes. Stay tuned to Genomics Revolution.